Okay, good morning, everyone. So welcome to the seminar. So as usual, we are on a webinar mode. So those on Zoom, you can report any technical issue into the chat. And please use the Q&A tab for asking any question, and we will allow you to interact with the room at the end of the seminar. And with that, I thank Laura for introducing to the speaker.
Thank you. Hello, everyone. Thank you for coming so numerous to this SEM seminar. Today, we have the great pleasure of welcoming Claire Mérot, who will be talking to us about her work on structural variants. Claire, briefly, she did her PhD at the Natural History Museum in Paris. There, she worked on divergence and mimicry in different heliconius butterfly species. And during that time, she also went to Peru to do some sampling and to try to breed these butterflies. After that, she went to Canada, where she did a postdoc at Laval University in Quebec. And there she stayed 6 years, and she worked on inversion polymorphisms in seaweed flies. And now she's back in Rennes, where she is going to continue her great work, and she will tell us about it today. Thank you.
Thank you, Laura. Thank you very much for the invitation. I'm to stand here, right? So yeah, it's a great pleasure to be with you today, and I will try to present you. So some of the research I did here, the results I feel they are already a bit old, but at least they're already published, and they come from what I did as a postdoc at Université Laval in Louis Bernatchez group. And most of that is the basis of the research I want to keep going. So I will finish by presenting you the new project that I'm starting. So overall, I wanted to highlight the significance and the role of structural genomic variants, and we'll present two empirical examples. So I guess as most of you, we are interested in the origin and evolution of biodiversity at different levels, both trait diversity, species diversity, and how that translates into ecosystem. And the basis for that is a genetic variation, and some people can call that sometimes the fuel for evolution, because it's the variation on which selection will act. And we are generally interested about how this genetic variation interact with ecological components and in shaping the evolution of diversity. And if we zoom in on the genetic variation, we know that genome varying sequence, we can becoming quite straightforward to study single nucleotide polymorphism, but we also know that genome varying structure. I've put here a few examples in which you have a pieces that's missing or that's inverted, so that's different type of structural variants, and there are some that are even more complex. And why would these variants matter? Why could they be different for evolution? So first of all, they can have important direct effects, sometimes simply because they are long, so they can be more likely to hit something functional than a nucleotide substitution. They can also have direct effect on gene expression dosage. You can sometimes predict that the more copy of a gene, the more it can be expressed. Roughly. And in terms of evolution, contrary to a nucleotide that may arise with error, and we kind of know well the mutation rate, the mutation rate of structural variant is possibly more variable. For example, transposable elements, they can make appear very quickly insertion or deletion or with birth sometimes. So that kind of change the dynamics of the apparition of novelty in the genome. And I've put here a few examples in which a structural variant is a large effect variant that impact important phenotype for adaptation and sometimes a rapid adaptation. They can also have indirect effects, both on gene expression, for example, by decoupling the gene and the regulation region. They can impact the chromatin condition, the 3D organization of the genome too, and the epigenetic marks. So overall, having a more indirect effect. And properties that some structural variants have that have attracted a lot of attention is that they can reduce recombination. So here is the example of an inversion. And so when the sequence is in the same order, meiosis and recombination occur normally. But in heterozygotes, where the arrangement is in different direction, so crossover do not behave super well, or they can result in unbalanced gametes. So overall, we can say that there is reduced recombination between these two arrangements. And this is very important for evolution and for adaptation. If you consider, if you think about how selection is multidimensional with a complex environmental variation that can vary with different sources and different scale, organisms can get locally adapted through selection, but this is also homogenized by migration. And in such a case, it's known that recombination will matter a lot. So recombination can be very benefic because it can generate new combinations, it can provide more modularity, for example, adapting to new conditions. But it can also break allelic association. So in some cases, a combination of ecological and genetic factors would favor reduced recombination, which may oppose migration and protect combination of co-adaptive. So that's why it's predicted that inversion, for example, could be important for local adaptation in the context of hygiene. So maybe it's not so surprising that we are finding more and more examples in which there are coexisting ecotypes with different traits in which one inversion underlies complex traits. I'm sure here you know well the Numata example with Mathieu showing you that. And it can also be important for speciation. So on the left, it's example at intraspecific level, and on the right, example in which chromosomal fusion or chromosomal inversion are associated with reproductive isolation. For example, because this can tighten reproductive barrier together. So we have been seeing a large effect, ecologically relevant structural variants, but I think there are many more of that. And the question we can start asking is to what extent what we see is just the tip of the iceberg, and there are many more structural variants that may not be adapted and just be neutral or largely deleterious, or do some of them are specifically exception, and what are the properties that would make them particularly different or important for speciation or adaptation? And I think we are at a good moment to start looking into that. we have been knowing the structural variants for a long time, thanks to cytogenetics, for example. We knew they were widespread through taxa, but we were focusing on few big structural variants. With the progress of genomics and bioinformatics, and I'm showing here an example of different methods, we can detect and genotype many, many structural variants. And what we realized is that there is much more genetic variation than we thought. They would cover 3 to 10 times more base pair than single nucleotide polymorphism. So that means they underlie a large part of genetic diversity that is still a bit overlooked. So with this large effect SV, we know they are important in adaptation. That's what evolutionary ecology has been showing. Thanks to medicine and bioinformatics, we know we are now able to catalog structural variants, but then we want to go beyond catalogs. And with population genomics, evolutionary genomics, generally we are trying to link genetics, phenotypes, and ecology, but we are mostly doing that on SNPs. And what I guess I like to do, and it's arising everywhere in the community, I guess many of us are doing that now, is trying to use this framework that's developed for SNP and maybe expand it to more generally ask how different genetic variants underlie evolution and this link between genetics, phenotypes. So I'll start with the example of chromosomal inversion in the civet flies. This is this little fly here is the Celopa frigida. It's living on the pile of rice bed, the seaweed that accumulates and that are really smelly on the seashore. We have it in Brittany. It goes up to Norway, and it's also in Canada. So that's where I did most of the research here. So we were interested in that species because of a very large polymorphic inversion that's been studied in the 70s and 80s. This inversion has two main arrangements, alpha and beta. So you can consider them as super allele of a kind of super locus. And this was known in Europe, not in Canada. And we knew the link with phenotype, so you can notice the large effect on the size. The homozygote alpha-alpha are 3 times bigger than the homozygote beta-beta. And when we kind of returned to the system with Maren Wellenhofer and Emma Verdam, we were interested in looking at this polymorphism and the genomic feature behind the inversion. And what you see here is the sampling population in Eastern Canada, and the different color are the proportion of the different genotypes. And you can note that it's super polymorphic. Like everywhere you will find the different genotypes. And in most populations, we have an excess of heterozygote, suggesting that there may be balancing selection going on and some advantage. So to better understand that, I've raised them in the lab, doing many generations. I've been genotyping eggs and adults. So this was cool because we could separate fitness into the survival component and reproductive component. And what we observe is that the beta allele gives an advantage for growth. They tend to survive better and get faster to adulthood. But the alpha allele gives an advantage for reproduction. The female will have more eggs and the males will make more. So overall, this is a classic life history trade-off, but here it is underlaid by the inversion and results in a very strong overdominance. So overall, there is this balancing selection that emerged both from higher survival of heterozygote, but also from this trade-off. And something kind of interesting here is that the relative fitness advantage of the different genotypes also depend on the ecological condition. If you think about it, like on the seashore, the seaweed, they can be of different quality and they can stay longer or shorter time, depending on storms and tides. So sometimes, at some places, it's a more advantageous strategy to develop fast before the right bed is taken, even if you reproduce less. But sometimes it's more advantageous to take the time and then be a good adult with good reproduction. So I guess that's more or less the kind of story we can imagine around that. And we've been testing that with models, and this also fits with the relative frequency in the different populations. And I believe there is some fluctuation over the year that we are trying to measure now in Britain that I can go very often to the field, please. Okay, so to make it in this framework, in this link, we found this inversion underlying life history trait with trade-off between survival and reproduction, some over-dominance, some non-associated mating, but hard to show. And the temporal and spatial variation in the quality and availability of the habitat tend to even enhance this polymorphism. Now, what about the genetics? To get into that, we opted for a strategy in which we did low coverage whole genome sequencing. So we are thinking about pool sleep to have good information about allele frequency. But with this method, we did one library per individual. So that means we can also return to the individual information. So at the level of population, we have 100 weeks. At the level of an individual, we have 1 week. But because we can return to that, I can also group them by genotype. I can group them by sex, or I can group them by geography. So that gives us a lot of flexibility. So this is just the principal component analysis showing genetic variation across all samples. And so I'm sure you understand very quickly that the main driver of the structure here is the big inversion I showed you. And I like to show also the admixture plot because I think it's a kind of a one in tail. I used to work on speciation, and if I would have seen that with admixture, I would have said, oh, wow, there are 2 species and we have lots of hybrids. But no, of course. And if you look here along the genome, this is only 10% of the genome that is driving this pattern. So we really have to be aware of linkage disequilibrium. Of course, if you prune for linkage disequilibrium, you don't. If we look more specifically at linkage disequilibrium, you see this whole region of the inversion is in super high linkage disequilibrium. So this reflects what we can predict from an inversion, that there is not non-recombination between the groups. Maybe so, quite expectingly, the 2 arrangements, alpha and beta, were super differentiated. This is the FST statistic measuring the differentiation between the 2 homozygotes. There were high value of G-mass D in this region, reflecting likely this balancing selection and heterozygous effect. So all of this is quite classic for this kind of supergene or polymorphic inversion. What we are trying to do now, so with the team in McGill, we have been making 2 genomes, 1 genome for alpha alpha, 1 genome for beta beta. So this is the chromosome 1. This is the chromosome 1 of alpha alpha, chromosome 1 of beta beta. And you can see it's nothing as simple as a reverse sequence. It's something more weird, more rearranged. I don't fully trust the assembly at that region, so I want to improve that. We did IC to do that. And there are some pieces that are more divergent and others more similar. So this suggests it is a nested inversion. And it may evolve a bit like a sex chromosome sometime, or they can have different strata inside this kind of old inversion. So something I want to dissect with the middle. So to summarize here, we confirm there were these linked haplotypic blocks that are highly divergent, rearranged, and they include hundreds of genes. So this kind of big locus is sometimes seen as a kind of an all-in-one solution, while it's a big block that underlies many traits that underlie complex phenotypes. And this link with life history effect, as some people suggesting it could be kind of better gene strategy in terms of evolutionary efficiency. Okay, so now I told you we don't want to focus only on the tip of the iceberg and try to look into other structural variants. So using the same data, I scanned the genome to look for other signature of non-recombining blocks of haplotype. So it's splitting the genome in pieces of about 100 SNPs, and look if we have this kind of pattern of PCA with 3 groups, and if the pattern is repeated across a few like hundreds of kilobases. So we are able to detect big blocks that are non-recombining and divergent. With these methods, we found the inversion, so that confirmed the method. And On chromosome 4, we found 3 inversions, one that is independent of the 2 others, and that is consistent with what has been described by cytogenetics in Europe. This was in Canadian population. And we found a region of low recombination that I'm not sure what they are. There could be some rearrangement, there could be methods in centromeric region. So this is something I want to also dig into more now. And so we were interested in seeing how those inversions will relate to environmental heterogeneity. Could there be under selection? And our sampling span 10 degrees of latitude. So for example, I put you only one ecological variable where we look at association between climate and different SNPs. So this is done with bypass. And overall, we found lots of outlier SNP associated, and a lot of them were inside the inversion. So of course, there is linkage equilibrium, so that's kind of something you would expect. But still, there were quite a lot, particularly in the inversion on chromosome 4. And now we can consider that another way. So I told you this inversion can be a big locus, and it is also a sequence. So before I was looking at SNP inside, and now I can look at it as a locus. And this represents the frequency of the inversion as a function of the latitude. So you can see the first one here. I think I lost the mouse. So on chromosome 1, it's polymorphic everywhere with no relation. For you, not for the one. So yes, here on the chromosome 1, it's just polymorphic everywhere. And the other one, you can see this line, meaning that the inversion is more frequent in the south and less frequent in the north. So this kind of fit with the association of having SNPs and suggests adaptation to climate or things that vary is now. So for this inversion, we went the other way around, like starting with the genetics, we detected those blocks and found that they were associated with ecological variation, more specifically latitude and temperature. And now I've been looking in European data, and in the European data, we found the same inversion that tends to have the same climb in the same direction. We were discussing yesterday if that could be some support for an adaptive effect. And what we are trying to test now is find if there are phenotypes associated with this big inversion. So this is the work of Léa Nicolas, who is starting her PhD. And what she measures here is supercooling temperature. So that means these insects, they go up to the north, Svalbard, so they're super resistant to cold. So we wanted to measure at which temperature they freeze. And when they freeze, they release energy. So that's the difference. And it's quite impressive because some flies, they won't freeze before -22. So they have something that protects them from freezing. So Léa will keep working, testing different phenotypes, for example, development time at different temperatures. So to summarize, in this species, we found kind of contrasted dynamic with an inversion that is a kind of balanced supergene that is super polymorphic with lots of heterozygotes, and 3 clinal inversions for which we suspect some relationship with adaptation to temperature. And both of them vary locally and could be involved in this adaptation to heterogeneous habitat, despite a very high connectivity. I didn't show you that, but there is really almost no genetic differentiation across 1,000 kilometers. And because this is maybe still the tip of the iceberg, I'd like to contrast this inversion to more inversion polymorphism. And the problem with the scan with the PCA is that we only detect inversions that are divergent and polymorphic enough. We are not really random in terms of inversion detection. So I believe to get into the full distribution of inversion, we need to do more direct detection using long read or other methods. So this kind of method I've been testing, not on the fly, but on the fish system. So now my second example, I wanted to tell you about structural variants and in the case of the speciation of the lake whitefish. So this lake whitefish is not one species, but two species in Canada with Northeast America. One species is a dwarf species, which is in the linentic part of the lake. And the other one is called normal, it's bigger and it's benty. And they differ in what they eat, how fast they grow. They have lots of ecological differentiation. And the first genetic data that they had on that allow to reconstruct the demography. And the best scenario is that it's likely that during a period of allopatry, they have been diverging. And upon recent secondary contact, there has been some ecological displacement with this, the Acadian glacial lineage becoming the dwarf species. So this species have been studied a lot in my former lab, including by Pierre Alexandre, and they knew a lot about all of that. But what they didn't have at that time is the genomes. So we wanted to use new genomic data and the full reference genome to get into the genetic architecture of species differentiation. And more specifically, I want to test if we can characterize structural variants. So to do this, I reused data from Clément's PhD. What he did have is so short reads for 32 samples that are separated between about 8 of each species coming from 2 lakes. The 2 lakes are kind of the same repeated story of differentiation. And they did long read on 1 sample of normal, 1 sample of dwarf with Oxford nanopore. The team of Sigborn Yen in Norway did amazing work at assembling the genome, and we got good genome assembly. And with this genome assembly, I was thinking that would be the best data to detect structural variants, right? You put one genome against the other, and you will see everything that is different in terms of structure. So I've tried that. There are different tools that one can use. I've used 3 tools and try to be quite conservative and ask the SV to be detected by at least 2 tools out of 3. It's a way to remove some false positive. Maybe not the best. And we detected about 90,000 structural variants in the dwarf relatively to the normal. But these individuals are just represented by 1 haploid reference, but they are diploid. So we wanted to come back to just the heterozygosity. So what Christina did is she took long reads, mapped them on the genome assembly, and we used that to detect structural variants with long reads. And we got a lot more. Those long reads are quite powerful to detect structural variants, and possibly also because we have heterozygote structural variants in the dwarf and in the normal. But again, that's just 2 samples, so I'm not sure how much it tells us about genetic diversity. So I thought it's better to come back to the 32 samples, right? It works. You can do, of course, SNP calling, and we can do structural variant detection with different tools. But we have much less structural variants, which in one way are interesting because they are population level variability. But in another way, that reflects how short reads do poorly when one wants to detect structural variants. And possibly it was even worse here because the coverage was really low. So in the end, I had different data sets, different catalog of SV that I tried to merge, and the overlap is limited. One of the reasons is that it's difficult to merge. And the main reason is that the different tools detect different types and different lengths of structural variants. So all of that to say that it's still not straightforward to detect structural variants, and one has to be very careful with the data sets one takes. And for example, it's hard to compare a study done on short read and one done on long read. You may have completely different things without any of them being completely wrong. It's just they work differently. So what I decided to do is like take all of them, try to represent them in a graph. So that's the new idea of how we can represent the genome. Because the more we see the structural variant, the more we realize that having just one reference per species is a bit limiting and does not capture the diversity. So there are all this new set of tools to make graphs it's called pangenome sometimes. So thinking in a graph, it's like when it's similar, there's just one track. And when there is a big divergence and insertion, we will have, for example, a loop with one reference path and an alternative path. So that's what we represent with T. And the beauty of that is that you can map on the graph the short read. And if the short reads map all on the reference path, you will say, okay, it's homozygote for the reference allele. And if you have some reads mapping on both of them, you can infer it's heterozygote, etc. So mapping on the graph allows us to genotype structural variants and then study those structural variants. So in terms of population, with the same filters, we add, of course, much less structural variant than SNP. These variants are less frequent, but because they are longer, they would cover about 5 times more of the gene. Then, was it worth it? This is the PCA based on structural variant on the left and SNP on the right. And yeah, when I did that, I said, oh, I'm wrong. I didn't take the good data set. But not really. It's just that there is so much divergence between the two species in blue and red and between the two lakes that I think with a few microsatellites, I will have the same, or a few SNP. So it's just to say that depending on what result we want, what question we ask, we don't need 12 million SNP. But then along the genome, we were interested in the pattern of genetic differentiation. So this is FST statistics between normal and dwarf in one lake and the other, with SNP and with structural variants. So yeah, I'm not sure we can say a lot about this, except that the differentiation is widespread in the genome. And we were looking at maybe what can be in common between the two lakes or just one lake. Can't say there is like more parallelism or difference. So yeah, these two species are really super divergent, and I wouldn't interpret any pixels, anything like barrier or gene or whatever. The one thing interesting I wanted to show you is that structural variants, and a third of them were associated with transposable elements. And this is due to the way transposable elements they do this kind of cut and pass and copy and pass, so they create insertion and deletion, and sometimes they can also favor inversion, so they can be at the breakpoint. So what we did here is simply look at the sequence and annotate structural variants that contain transposable elements or do not contain that, and some that will contain more repeated elements. So on the left is the pool of structural variants, and on the right, it's structural variants that are outlier in terms of being more differentiated in frequency between the two species. And we see an enrichment for many T families. So yeah, I'm wondering, like, there could be some bias in it in terms of possibility of detection of variation frequency. But one scenario we could speculate is also that because T tend to have a faster mutation rate, and the divergence between two species is quite recent, it could be that the insertion or deletion created by T like occurs differently in the two populations, in the two species, and that's the traces of that we see. And this kind of hypothesis fits with what has been observed in the hybrids. So this is a completely different study, but what they found is that the hybrids and the backcross, they do not survive very well. And this is possibly due to transposable elements that are kind of reactivated, and there is this hypothesis of genomic shock. At least what they observed is that in terms of methylation, so usually methylation would silence transposable elements. And each species would be good at silencing its own T. But then in hybrids, it may be that they are not so good at silencing the T on the chromosome coming from the species. So that's what they observe here, is that there is like the methylation patterns are completely different and tend to be less methylated than the pure species. So I think this kind of process is interesting in terms of speciation and in terms of thinking about structural variants, because they can give you like a genome-wide syndrome in that case. And this is something that is emerging in other types of variants. Very quickly, I wanted to show you here. Very recently, we have been thinking and writing about structural variants and speciation with Mathieu here and a group of colleagues. And structural variants have been for a long time involved in speciation. This is the classic hypothesis of chromosomal speciation for two reasons: because of recombination, coupling barriers, I mentioned earlier, or because of direct effect on meiosis. But in the literature, it's now emerging that structural variants have different properties, just their length, their effect size, different dynamics, like we did for T, and they can have widespread impact in the genome. So I've mentioned the deregulation of T, but there are other genome-wide effects. For example, in this mouse, they've been looking at 3D organization of the genome, and with chromosomal fusion, they realized that it changed the 3D organization, and some fusion can lead to more fusion of chromosomes. There are more and more examples that chromatin accessibility could be related to SV density, and cases of burst of T that could be associated to speciation. So I think it draws a more complex picture in which structural variant can lead to more structural variants, and this kind of genome-wide effect could contribute to speciation. So, yeah, to summarize, here I wanted to tell you a bit about the role of SV in the evolution of biodiversity with an example of intraspecific ecotypes, an example of speciation involving also structural variants, also about the methods that we can use, both direct detection and indirect detection, to study that, and then how we wish to integrate that better in population genomics by linking this genetic complexity to phenotype and ecology in evolutionary process. So now if I still have some time, I thought I would not present all stuff that coming first. I don't know what. Okay, so I'm a bit in between. So everything was done, and now this will be starting in this winter. It's a new project, and I've been lucky that it gets funded by Europe. And I want to keep working on the role of structural genomic variants, and in this case, it's more specifically about eco-evolutionary processes and related to global change or rapid changes in conditions. So it stems from the observation that we discussed already, that considering structural variants change the magnitude of variation that we capture with a broader fraction of the genome covered by variants, that these variants have specific qualitative difference. So I mean, shifting from SNPs to SV will bring like quantitative and qualitative difference in the way we consider genetic variation. So this impact on recombination can make adaptive cassettes, supergene favorable, but we also know that this will limit the formation of new combinations, which can be important when the environment changes. And it also limits the purging of the nucleus. Maybe it's not so good to have this inversion everywhere in the gene. We already mentioned that the idea of the speed of evolution that could possibly be faster. Like, for example, for duplication, it was said that their mutation rate is higher than SNP, but selection is also higher against them. So the evolutionary rate is not comparable to what we can infer from SNPs. And this can be very important for rapid adaptation. I put here the example of insecticide resistance that you know. And also they are known to be more likely to be deleterious. And this starts to be a concern for threatened species, for example. This is the kakapo parrot. And they start to find this big rearrangement that could be not so good for fertility. So overall, more variation, properties that impact the evolutionary process. So we can make the hypothesis that they have the potential to favor, but also limit adaptation in rapidly changing environments. So this is what we would like to test, is whether the SV play a significant role in the evolutionary response to global change. And well, this I said earlier, that we do have company examples, but no systematic test. We can catalog SV, but now we want to do more. And we want to use population genetics also on S. So what would I like to do with that? First, there is a large part of the project that is experimental, trying to have an integrative approach, both studying natural population and experimental population. And this is fairly easy to do with the seeded fly model because it has a very short generation time, 8 to 15 days, can be raised in the lab, you can control the conditions. In nature, we know the habitat. It's always a dry bed on the beach, but they span a very large gradient on the European continent, but also in Japan, East America, and West America. And in Europe, there is also a related species, Pilipes, that is shifting north. Over the last 20 years, I think it shifted for 3 degrees of latitude. So I think this species is really in expansion. It's start to outcompete the Frigida in Britain. So we're interested to look at this dynamic that I think happening to kind of a few species, many species at the moment with the change in temperature. And the key to be able to do that is to have SV in large data. If we are able only to detect SV in 2 samples, because we can only do long read, that won't work. So thanks to a Montpellier platform, we hope that I could be trying to do haplotagging, linked read. So the idea is that before we break the genome to sequence it, we would tag the long molecule. So then when we can reconstruct in silico which molecule, which little reads come from the same long molecule. So this is great for phasing, and this should be great for detecting longer structural variants. We will also do some long reads, but again, not so many samples, because haplotagging can be done on many samples. And then using new bioinformatics tools such as the graph I mentioned earlier, and new data, because they are both Darwin Tree of Life and ERGA are providing new reference genome for many species that provide an opportunity to the high quality of the genome at chromosome level is great to be able to look at structural variants. So first, let's see how SV vary in natural population. To be able to genotype structural variants in many populations, many flies, can help us get into the evolutionary rate, the distribution of fitness effect. And I'm particularly interested to focus on one property, which is the impact on recombination. Because this is known for inversion, but could also be for other structural variants. So we hope to be looking at that in natural population and also experimental crosses. Second, I mentioned to you the species that is in expansion. So we could expect that there is some expansion load that's being documented. And in the south, this one is really becoming fragmented. So there also could be a reduction of genetic diversity and possibly more drift, more deleterious variants staying around. And because SV are expected to be more deleterious, I want to kind of look at this interaction between the different type of variants and the demographic dynamic. And comparing, so Pselopa frigida has many large inversions, and Pilipes is supposed to have none. At least in cytogenetic, they never found any. So also these 2 species can contrast 2 situations. Third, so I mentioned this parallel gradient, and there is this hypothesis that some properties, for example, inversion reducing recombination, make them more likely to be picked by parallel adaptation. And also to be involved in rapid response. So this is something we can test experimentally in the lab. It's kind of easy to make many generations, controlling temperature or condition. And last, I like to do like something broader. And the idea here is that with this new reference genome available, we have just one genome per species, but we have exactly the same data and the same way of processing it. So it's a great data set to be able to compare between species the variation, but of course we don't have diversity, we have more heterozygous. But with that, we can then test different predictions, for example, looking to Leontine's paradox and impact on that, or relate the diversity of inversion in the species to a life history trait or the way recombination occurs, for example, sex bias recombination. And I'm sure there are other hypotheses in which ecological or life history or, I don't know, developmental mechanisms affect how likely they are to keep or to make appear different kind of structural variants. So if you have ideas on that, I'm happy to hear. And hopefully later, more and more population data set will appear for structural variants, possibly with haplotagging or with more long read. I know some groups are doing that. And in the future, it will be great to be able to compare what we observe with this more comparable data set to have population information about structural variants. So all in all, there is a lot of things. It's not started yet, so I'm very happy to hear any comments, suggestions, how we can do it, how we can make it better. And I'm doing a shameless promotion saying if you're interested in any of that, I will be recruiting at all levels. And with that, I'd like to thank the many people that have been involved all along this study, and thank you for your attention. Happy.
Thank you so much, Claire. Do we have questions in the room first, and then after we can ask on Zoom? Okay. Well, okay, yeah, Nicola.
So you said that you start by touching the tip of the iceberg of structural variants, and now you are trying to have the full pictures. But if you think about SNPs, do you think we have the whole picture, or are we also just touching the tip of the iceberg?
Yeah, that's a good question. Well, I guess it depends. Because what I felt about structural variant is that it started to change over the last few years. But before, we were more looking at the phenotypes or very different ecotypes, and then grasping one structural variant. So of course, we are more at the tip than what we do with SNP. Because with SNP, we can do a bottom-up study. We have all SNP, and then we ask, do they matter for that? Or doesn't mean we have all SNP and we know all of the effects, but more than what we did with structural variants. But it also depends on structural variants. Because, for example, transposable elements, they have been studied a lot for a long time, or duplication. And then the focus was less on one phenotype linked to one variant, but more the distribution of TB across the genome. So maybe in that case, you are more in all the iceberg.
Thanks for a great talk, Claire. I was wondering, I'm curious to hear more about the pan-genome graphs. I don't know if you have any pictures or you can tell us a bit more about them. They just sound very interesting, and I'd love to hear more.
Yeah, I don't think that. You mean how they are done, or? Yeah. So the idea is that instead of having one reference per species, why not start to have several well-assembled species? So then how do you handle that? You do a map on one of them or the other one. So the idea of the graph is to be able to, because when they are super similar, you can have just one path. And when they have this structure that makes them different, that will make loops in the graph, sometimes complex loops or branches. But it's a way of changing the representation, I think, for pangenome. Okay. By pangenome, I mean a collection of genomes. Does that answer your question?
Thank you very much. That's quite interesting to see a different perspective. And I was wondering, so we're trying to do similar things, but with fungi. And one difficulty might be how to separate the effect of anything that is nested within the structural variant versus the structural variant itself.
Yeah, I agree that that's always the question about is it because this piece gets inverted and disrupt the gene or something, or everything that's inside. One of the things that people do is look at the breakpoint themselves. Because if it's just everything is just repeated elements, you have less hypothesis than if you try to gene. And also what I've seen, and not to be crazy, is in Drosophila, they revert inversion. I won't be able to do that here because of the complexity. But yeah, and even inside the inversion, everything is linked. So you do a GWAS, you're just
no CRISPR-Cas, yeah. No CRISPR-Cas plan.
No, actually, people tell me it would be easy on that species because it lays a lot of eggs at the same time. Or maybe one day, yeah. But we need to have candidates for that.
Thank you, Claire, for the great talk. I have a question which is related to the previous one. In the end, we can assume that the fitness effect of an inversion, for instance, is somewhat related to its mutational content. So how do you expect to establish links between this view at the structural variant level that you have to the amount and types of single nucleotide mutations it contains? Will you also explore this axis?
So I guess that more applied to the very big inversion than to small ones. Related to your question, I think for small ones, you can more easily get into direct effect. But yeah, for the big one, so what we add on that is also looking at gene expression, like to look at which gene are differentially expressed, but that doesn't mean we can get into all the states. So no, I'm not sure how to do inside the inversion. It's really a puzzle. Yeah. What we can, so one thing is that inside each haplotype there is variability. So perhaps with that we can reconstruct somehow the history of each haplotype and some genetic effects.
Maybe based on SNP divergence.
So very basically, just based on DXY and kind of molecular clock. It's around 200 or 300,000 years for the big one on chromosome 1. The one on chromosome 4 is a bit longer. But I'm not sure how much I trust this number.
Sure. I was also thinking of the T's that you find in regions that seem to be, let's say, outliers at the right of the distribution. Do you see also that they are younger, for example, so they could correspond to more active elements?
You mean in the whitefish?
Yeah.
So we don't really, well, we don't really think they are active at the moment.
Mm-hmm.
Well, there are some peak of SV that corresponds to T. So there has been really moment, probably of birth of TC1 mariner or this kind of thing. But there were no differences, for example, between two species in the relative proportion of the different families, which is something I was expecting. Maybe I don't know enough about T, but I was thinking maybe one family is active in one species and one family in the other one. There were no significant difference of proportion.
Maybe it's a recent divergence also. So.
Yeah, exactly. And related to the inside inversion, there are lots of T inside the inversion. So perhaps something also that could help date or think about the date, but it's also known that they are less able to purge this inside the inversion.
Yeah.
Don't know what the cause and consequences. Yeah.
We can talk. Sorry, maybe just one last one. I was curious because you say, so you are going to sequence a bunch of genomes, generate the graph, and then realign the short reads on that. How do you plan to, control for maybe a certain bias that could be generated by the fact that you are only going to type the for example, the you have in genomes?
So that's not really the plan for the CV5 for the new project. Because first with haplotagging, I'm not sure how much we will get into pangenome, or maybe we do this with long reads. But then, of course, that's the bias that we can only genotype what we detected. And of course, you don't get into the very rare variants, or you only have the rare that are present in one sample. So it's not so great for the distribution of fitness effect or question of expansion node. So I would be afraid of that. So maybe it's more de novo discovery for this kind of population analysis.
We still have time for a question. I am.
Maintain the policy. Those is more important. Morphism. Do you whether appeared or did they here?
You're talking about the. Okay. So the question, and I guess it addressed the first inversion of the Celopa frigida, right? The big one. The question was, what's the relative importance of overdominance and local adaptation in the maintenance of the polymorphism? So yeah, we were interested into that, and we did a kind of a model, like very experimental model, just making flies reproduce in the computer, individual-based model. And that way we could add, separate these different parameters. So we could remove, for example, the intrinsic overdominance, so the fact that heterozygotes are slightly better than both others in just the vibe. The other aspect of overdominance that's bigger is resulting from the trade-off adaptation. And this trade-off, the advantage of the trade-off of each genotype really fluctuates depending on the environment. So I'm not sure we can quantify what is local adaptation, or maybe we shouldn't call that local adaptation. It's more than locally, at one moment of the year, this genotype will do better. Maybe 2 months later, it's different. So all of this creates fluctuating, like heterogeneity, that enhance the balancing selection. If we put no heterogeneity of the habitat, I think the trade-off is the range of parameters in which polymorphism maintained is smaller. I'm not sure that answers your question about the relative importance, but that's what we explore at least. And then the second question was, what happened at the origin of this inversion? I guess, yeah. And that's a big question for all these old and big inversion. And it's hard to imagine how they establish in the first place. Particularly if this one is nested, maybe we had intermediate stage, I'm not sure. What we would like to do to better understand is so there are other species of celopids in which they just have big males and regular females. And other species in which they have small males and small females, but not this polymorphism. So we want to look broader in the clade about this distribution of phenotypes and possibly the presence of the inversion, to kind of try to know if this inversion is really specific to that species, or if it's something that could be like possibly introgress or some gene. So I have no idea about the origin of this inversion. I don't even know which allele is derived and which one is ancestral. Perhaps that's the first thing to look into, right?
Maybe perhaps to follow up to the Mark's question, one way to look at that would be to experimentally investigate whether the fitness of your two homozygotes vary with temperature. So presumably you lose overdominance when you are really cold or really warm, because the inversion is no longer polymorphic. And then the overdominance is just occurring at intermediate temperature. And so perhaps you could investigate this experimentally.
Inversion on chromosome 1, we didn't see relationship with temperature. Sorry, maybe because of the two. So the one on chromosome 4 has this climb with temperature, and this one does not seem to have any excess of heterozygotes. And the one on chromosome 1 is the one with heterozygote excess and balancing selection. And there is no real climb, exactly. So no effect on it. What we see, though, is association with what the substrate is. in nature. So that's what I tried in the lab. I had Fucus and Laminaria, and I was raising them on 2 substrates, and we saw no difference. And I think one of the reasons for that is that I was giving them all the time they wanted. So I saw alpha taking over very quickly in 5 generations to frequency that never happened in the wild. So we were really puzzled at that moment. So that's when we look into eggs versus adults. So that's why I think there's something with the habitat, how long it is and the quality of it. But it's, yeah, experimentally, everything we tried, it didn't really show up. What is known though is the density. If it's very dense, the heterozygote have a very strong intrinsic advantage. And at lower density, it's more or less erased. So depending on the ecological condition, the intrinsic heterozygote advantage varies. It's really intertwined all this factor together.
So before we all go to lunch, we just had a small announcement to make, which is that the same organizing committee is organizing a new conference, which will take place next week on Friday. The aim is to gather the community at the beginning of the university year. So exceptionally, it will take place in the afternoon at 3 p.m. and it will be in the amphitheater on the other building where you used to go as well. And so this year we will have the pleasure to listen to Frédéric Villard, which will be here. And the idea is also to have drinks and big apéro right after to just gather together for this new year. And so it will be funded by LabEx, and so around 4:30 we will also have drinks. And we hope to see you all there next week. And with that, thanks very much, Claire, for this very interesting talk.